Protein synthesis. We completed replication, transcription. Today we are moving to discuss this. What are protein synthesis, or otherwise called as translation? Translation. Okay. Translation is the mechanism by which triplet base sequence of a mRNA guides the linking of a specific sequence of amino acids to form a polypeptide on ribosome. Simply, it is the formation of polypeptide chain from mRNA. Okay, protein synthesis which occurs over ribosomes. Ribosomes are required for translation. Then, it consists of ribosomes. So, what are the machineries which is required for protein synthesis? It requires ribosomes, amino acids. Okay, ribosome, amino acids, tRNA, ribosomes, amino acids, tRNA, then rRNA, tRNA, mRNA, rRNA. Okay, these are the essential raw materials which are required for the protein synthesis. And amino acyl tRNA synthesis. Okay, we will discuss about what is amino acyl tRNA synthesis. So these are the raw materials which is required for what? Translation. Then come to the steps of protein synthesis. The first step of protein synthesis is the activation of amino acid. Activation of amino acid. Activation of amino acid. 20 type of amino acids are participate in the protein synthesis. 20 types of amino acids are participated. 20 type of amino acids are participated in the protein synthesis. Amino acids react with ATP to form amino acyl AMP enzyme complex, okay, which is also known as activated amino acid. What is amino acid? Amino acid react with energy molecule, that is ATP, to form amino acyl AMP enzyme complex. Amino acyl AMP enzyme complex. Amino acyl AMP enzyme complex. How it is formed? When 20 different types of amino acids are involved in the protein synthesis. When amino acid combined with ATP, to form amino acyl AMP enzyme complex. Okay. So we will write amino acid plus ATP combined with the help of an enzyme called amino acyl tRNA synthetase. With the help of an enzyme, amino acyl tRNA synthetase. Amino acyl tRNA synthetase. Okay, so it will give amino acyl AMP enzyme complex along with 2 inorganic phosphate. Okay, amino acid. So this reaction is catalyzed by this specific enzyme, amino acyl tRNA synthetase. There is a separate amino acyl tRNA synthetase enzyme for each kind of amino acid. Okay, separate amino acyl tRNA synthetase enzymes for each amino acid. So this is the activation of amino acid. First, we activate the amino acid by combining the ATP, which give amino acyl AMP enzyme complex plus 2 inorganic phosphate in the presence of amino acyl tRNA synthetase. Okay, then come to the next step, that is the charging of tRNA. Charging of tRNA. It is the second step. Charging of tRNA. Specific amino acid, that is specific activated amino acid, that is the charged amino acid is the activated amino acid. Okay, specific activated amino acid is recognized by its specific tRNA. So specific activated amino acid is there for a specific tRNA which is recognized by them. Now, amino acid attached to the amino acid attachment site of its specific tRNA and AMP and enzymes are separated from it. Okay, so what happens when the amino acid attached, that is the activated amino acid attached to the word amino acid attachment site of the tRNA. Okay, amino acid attachment site of the okay. So this is the amino acid attachment site. When the activated amino acid attached to the amino acid attachment site of tRNA, what happened means the AMP and enzymes are separate from it. Okay, the AMP and enzyme complex separates. Okay, separates from it. Okay, so what is the reaction? Amino-acyl AMP enzyme complex. Amino-acyl AMP enzyme complex plus specific tRNA molecule. Will give amino-acyl tRNA. It is amino acid binded tRNA. Amino-acyl tRNA plus the AMP and the enzyme complex will what? Separates from the amino acid AMP, and that is from the activated amino acid. The AMP and enzyme will what? Separate. AMP plus enzyme. Okay, AMP plus enzyme. So this is the reaction which is taking. Amino-acyl tRNA complex is also called charged tRNA. Amino-acyl tRNA also called as charged tRNA. Charged tRNA. Okay. Now this amino-acyl tRNA move to the ribosome for protein synthesis. Now it is ready to move to the ribosome for the process protein synthesis. Okay, then come to the third step of protein synthesis, that is initiation of polypeptide chain. Initiation of polypeptide chain. Initiation of polypeptide chain. Okay, so in this step, 30S and 50S subunit of ribosome, 30S and 50S subunit of ribosome, GTP, magnesium cofactor, charged tRNA, mRNA, and some initiation factors are required. Okay, so what are the machineries required for initiation of polypeptide chain? 30S and 50S ribosome. Then it required mRNA. Obviously, mRNA is required. Then charged tRNA is required. Then it requires what? Ribosome. Okay, ribosome is there. mRNA, charged tRNA. Then energy molecule. It is in the form of GTP. Then cofactor is required, magnesium. Then it required some initiation factor. Initiation factors. Okay. So what are the requirements? That is, what are the machineries? 30S and 50S RNA, mRNA, charged tRNA, GTP, that is energy molecule, guanosine triphosphate, then magnesium as cofactors, and finally the initiation factors. Okay. In case of prokaryotes, there are 3 initiation factors. In case of prokaryote, 3 initiation factors are there. We will name as IF1, IF2, IF3. In case of prokaryotes, there are 3 initiation factors are there, IF1, IF2 and IF3. Okay, and in eukaryotes, there are more than 3 initiation factors are there. Okay, in case of eukaryotes, there are more than 3 initiation factors are there. More than 3 initiation factors are there. Okay, the GTP and the initiation factor will promote the initiation process. The GTP and the what? Initiation factor will initiate or promote the initiation process. That is the initiation of polypeptide chain. In starting, both subunits of ribosomes are separate with the help of IF3 factor. Okay, at the beginning or at the starting, both the subunit, small subunit that is 30S and larger subunit that is 50S are in the separated form. Which is done by the IF3. Okay, so the function of IF3, they separate 30S and 50S subunit. 30S and 50S subunit. Okay, then in prokaryotes, with the help of SD sequence, in prokaryote, with the help of SD sequence, what is called SD sequence? Shine-Dalgamo sequence. Shine-Dalgamo sequence. Okay, Shine-Dalgamo sequence. In case of prokaryotes, the Shine-Dalgamo sequence and mRNA recognize the small subunit of ribosome. So, who will recognize the small subunit? Shine-Dalgamo sequence plus RNA. Shine-Dalgamo sequence plus the mRNA molecule. Okay, they jointly recognize ribosome. That is the small subunit. That is 30S. Small subunit is recognized by the what? SD. That is Shine-Dalgamo sequence as well as the mRNA. Okay. A sequence of 8 nitrogen base is present before the 4 to 12 nitrogen base of initiation codon on mRNA called Shine-Dalgamo sequence. So, Shine-Dalgamo sequence comprises 8 nitrogen base pair. 8 nitrogen bases which is present just before what? 4 to 12 nitrogen bases of the initiation codon. Okay. At the time of genetic code, we already discussed what are the initiation codon. Okay, initiation codon is AUG or sometimes GUG. In smaller subunit of ribosome, a complementary sequence of SD sequence is present on 16S rRNA, which is called anti-Shine-Dalgano sequence. Okay, in small subunit. Okay, small subunit, that is SD sequence, that is Shine-Dalgamo sequence is present in the Shine-Dalgamo sequence plus mRNA will recognize the small subunit. Okay, small subunit of the ribosome. How they are recognizing? A complementary sequence of Shine-Dalgamo sequence is present in the 16S RNA. Okay, 16S RNA, that is the small subunit 16S RNA, which is called anti-Shine-Dalgano sequence, ASD, anti-Shine-Dalgano sequence. Yeah, that is called ASD. ASD. Okay. So by seeing that anti-Shine-Dalgamo sequence, it is come and recognizing the ribosomes actually. With the help of ST and ASD sequence, with the help of this ST and ASD sequence, mRNA recognizes the smaller subunit of ribosomes. mRNA recognizes the smaller subunit of ribosomes, while in eukaryotes, smaller subunit of ribosome is recognized by 7-methyl guanosine cap. Okay, this is what? Prokaryote. In case of eukaryote, the smaller subunit is recognized by the 7mg cap, that is 7 methyl guanosine cap. Okay, 7-methyl guanosine cap is recognized the smaller subunit of ribosome in case of what? Eukaryotes. In eukaryotes, 18S RNA of smaller subunit has a complementary sequence for 7-methyl guanosine cap. Okay, so in case of what? Eukaryote, 18S RNA has a complementary sequence for 7-methyl guanosine cap. So by this complementary sequence, you can easily understand the what smaller subunit of ribosome. Now, what is going to happen? 30S subunit plus mRNA, 30S subunit plus mRNA, presence of some initiation factor and some cofactor, it will form a complex. Let's look how the complex will produce. Okay. The 30S subunit along with mRNA, in the presence of an initiation factor 3 and the magnesium cofactor, it will produce a complex called 30S mRNA complex. 30S mRNA complex is produced by the reaction of 30S, that is smaller subunit, plus mRNA in the presence of initiation factor 3 and the magnesium cofactor. Okay, so this 30S mRNA complex, is going to react with 30S mRNA complex. Okay, so this 30S mRNA complex is going to react with formyl methionine tRNA. Formyl methionine tRNA. Okay, formyl methionine tRNA means tRNA having the amino acid formyl methionine. Okay, formyl methionine tRNA means tRNA having the amino acid formyl methionine. Okay, so the 30S mRNA complex react with the formyl methionine tRNA complex to form, react to form 30S mRNA formyl methionine tRNA complex. 30S mRNA formyl methionine tRNA complex. Okay, when 30S mRNA complex react with the formyl methionine tRNA complex, it will form 30S mRNA formyl methionine tRNA complex. Okay, this tRNA attached with codon part of mRNA. This tRNA attached with the codon part of mRNA. Okay, this tRNA attached with codon part of tRNA. Okay, codon part of tRNA. It requires one GTP molecule is required for this attachment. Okay, this tRNA attached to the codon part of mRNA. Here an GTP molecule is required. Okay, so what will be the reaction? What will be the reaction? The 30S mRNA complex along with the formyl methionine tRNA complex in the presence by utilizing one energy molecule, that is GTP, in the presence of two initiation factors and a cofactor will give 30S mRNA formyl methionine tRNA complex. Okay, 30S mRNA formyl methionine tRNA complex. So what are the here one GTP is using, then the initiation factor IF3 and IF2 are the, then the cofactor Mg2+. Okay, so the 30S mRNA complex along with the formyl methionine tRNA in the presence of GTP, IF3, IF2 and Mg2, it will produce 30S mRNA formyl tRNA complex which attach the codon of the tRNA. Now, these are about the smaller subunit. Come to the larger subunit. Now, larger subunit of ribosome, that is 50S unit, joins the complex. When all these things happen to the smaller subunit, then only the larger subunit will come and join the smaller subunit. Now, the large subunit of ribosome, that is the 50S ribosome. 50S ribosome. Okay, the larger subunit, that is 50S, that is the largest subunit of ribosome, joins this complex. Okay, join this 30S mRNA formyl methionine tRNA complex. The initiation factor is released and complete 70S ribosome is formed. When 50S ribosome combined with the what 30S mRNA formyl methionine tRNA complex, when it is joined, what happen means this will release initiation factor, release IF factor, and what happened? Both 50S and 30S join to form a ribosome. Join to form a ribosome. Okay. Now, larger subunit of ribosome joins the complex. The initiation factor is released from this and complete 70S ribosome is formed. 70S ribosome is formed. Okay. Then, now we will discuss about the what major site which is present in the tRNA. Okay. In large subunit of ribosome, there are 3 sites for RNA, especially the tRNA. Okay, there are 3 sites for tRNA in ribosome. Okay, there are 3 sites are present in the ribosome for the tRNA. First one is P site. P site called peptidal site. P site called peptidal site. Then A site for amino acyl site. A site for amino acyl site, and finally the E site. Exit site. Okay. So P site, A site, and E site. P site is the peptidal site, A site is the amino acyl site, and E site is the exit site. Okay. Starting codon of mRNA is near to the P site of the ribosome. Starting codon. Please listen. Starting codon is of mRNA is near to the P site of ribosome. So tRNA with formyl methionine amino acid first attached to the P site. Okay. So tRNA having formyl methionine attach P site. Attach the P sites. Okay. Then starting codon of mRNA is near to the P site of the ribosome. So tRNA with formyl methionine amino acid first attached to the P site of the ribosome, and next codon of the mRNA is near to the A site of the ribosome. So new to tRNA with new amino acid always attaches the what? A site. Always attaches the A site. So if this is the ribosome, it has the what? mRNA. This is the large subunit. It has what? E, P, A. E site, P site, A site. First, the tRNA will come along with the codon and bind the P site. The first, that is the formyl methionine tRNA combined binds with the P site. Further tRNA along with the amino acyl tRNA will come and bind the A site. Okay. So what happened as a result? Then from here the tRNA come to the E site. And finally it will access. Okay. When the amino acids are released, then it will access to the it access to the nuclear plasmid. Like that, coming tRNA come and join the A site. The first amino acid tRNA will come and bind the P site. Okay, so as a result, what happen is here polypeptide chains are starts to form. Okay, that is the coming amino acids are joined one by one. Okay, so amino acids are joined one by one to form a polypeptide chain. This is what happened. This is what chain elongation. This is what chain elongation. Okay, so initiation of polypeptide chain over, then the next step is the chain elongation. Chain elongation is what I just tell you. Chain elongation. Chain elongation requires tRNA, GTP, magnesium as well as the elongation factor. So what are the machineries required? tRNA, then energy molecule GTP is required, magnesium cofactor is required, and EF, elongation factors are required. Okay, new tRNA with new amino acid attached at the A site of the ribosome. First of all, tRNA attached to the P site is discharged, so COH of the P site amino acid becomes free. That is the carboxyl site. That is the what 3' end will be free. So again, new peptide bond takes place between the carboxyl group as well as the P site amino acid. That is the amino group. Okay, this is what happens. E site, P site, A site. When a tRNA along with one amino acid come. Okay, suppose this is the amino acid, and I hope you know the structure of what amino acid. It has what carboxyl group, this is R, and this is amino terminal, carboxy terminal, and okay, so this is the structure of amino acid. When a tRNA will take this amino acid and come here, so the carboxyl group is free now. So again, one amino group of other amino acid can come with other tRNA and join this. This is called peptide bond. Peptide bond. Okay, bond between carboxyl group and amino group is the peptide bond. So peptide bond can be easily formed, and more amino acid can be joined one by one. Okay, more and more amino acid can be joined. So first of all, tRNA of P site is discharged. When tRNA is discharged, the carboxyl end is free. So another amino acid having the amino group can join these carboxyl end. Okay. Peptidal transferase enzyme include the formation of peptide bond. Okay, the bond peptidal transferase. Peptidal transferase is the enzyme which is involved in the formation of peptide bond. Formation of peptide bond in between two amino acids. Okay. Peptidal transferase enzyme induces the formation of peptide bond. In peptide bond, a 20S rRNA is also helpful. 20S rRNA also helpful in case of 23S rRNA is also helpful in case of peptide bond. This rRNA acts as an enzyme, so it is called as ribosome. It acts as an enzyme, so it is called as ribosome. 23S rRNA acts as an enzyme for this peptidal transferase activity, that is the formation of peptide activities. It is called as ribosome. After formation of peptide bond, tRNA of P site is released from the ribosome via E site and dipeptide attached with A site. Okay, I will look the animation video. If I get it, I will transfer to you, which will make you more clear. Okay, actually this replication, transcription, translation, animation video is more helpful you to understand what is happening. Next, ribosome slides over mRNA in 5' to 3' direction. So due to this sliding of ribosome on mRNA, new codons of mRNA is continuously available at the A site of ribosome. Okay, so ribosomes are moving through the mRNA. Not mRNA is not moving. Ribosomes are slides over the mRNA. Okay. Translocase enzyme is helpful in the movement of ribosome. Enzyme translocase. Translocase is the enzyme which helpful in the movement of ribosome. Translocase enzyme is the enzyme which is helpful for the movement of ribosome, and energy JTP is required for the sliding of ribosome. JTP is required for sliding of ribosome. Sliding of the ribosome. Okay. In elongation process, some protein factors are also helpful, which is known as elongation factor. For elongation, what some protein factors are also helpful, they are called as elongation factors. And if you see in case of prokaryotes, the elongation factors are, in case of prokaryotes, the elongation factors, they are mainly of three types, that is EFTU. Three types are there. Elongation factors are three types. First one, EFTU. Then second one, the elongation factor TS. Then third, the elongation factor G. These are the three types of elongation factor which is present in case of prokaryotes. Okay, and if you come to eukaryotes, you can see two types of elongation factors. Mainly two types of elongation factor. First one, EF1 and EF2. Eukaryotic elongation factor 1, eukaryotic elongation factor 2. Okay, so these are about what? Chain elongation. Then come to the last step, that is chain termination. Come to the last step, that is chain termination. Due to sliding of ribosomes over mRNA, when any nonsense codon, what are nonsense codon? UGA, that is upper umbel, umber. Occur umbel, umber. UAA, UAG, UGA. These are the termination codons or nonsense codons. When the ribosomes are sliding, this is the mRNA, this is the ribosomes are sliding through the mRNA. Suppose when during the sliding, suddenly the mRNA see this codon UAA or UAG or UGA, any one of these codons, the process will stop. Okay. So when during the sliding of ribosome over mRNA, when any nonsense codon is available at the A site of ribosome, then the polypeptide chain terminates. The linkage between the last tRNA and the polypeptide chain is broken by the three release factor. Okay, we can see release factor here. The linkage between the last tRNA and the polypeptide chain is broken by the three release factor, that is RF1, 2, 3. RF1, RF2, RF3 along with the energy molecule GTP. Okay, along with the energy molecules. Then peptidal transferase enzyme also catalyze the releasing process. Peptidal transferase enzyme also catalyze the releasing process. In eukaryotes, in case of eukaryotes, only one release factor is known. In case of eukaryotes, only one release factor is known. Only one release factor is known. It is known as ERF1. ERF1. Okay. So these are about the chain termination. Once the initial portion of mRNA is translated, it is free to engage into other round of translation. Okay. Suppose if the ribosome starts here and it slides over here and start codon came, so the ribosome will detach from the mRNA and the mRNA is free. Okay. this mRNA can again go and other ribosomes can bind, and again translation can be takes place. Okay, so this can be continuously happen. It is free to engage in another round of translation. This process can be repeated several times, so creating a structure of polyribosomes or polysomes. Okay, what is polysomes or polyribosomes? Polysomes or polyribosomes. Suppose you consider this is an mRNA 5', 3'. One ribosome come and bind, and this slides here and reaches here, and finally it reaches here. Okay, finally it reaches here. So the same time again another ribosome can come and bind the same mRNA, and when this ribosomes come and reaches here, again another ribosome can be come and bind the same mRNA. So this is called what? Polysomes or polyribosomes. So here new ribosomes will come and bind. Here the already the when the elongation, that is the translation done, the ribosomes will detach and it will release by this end. The new ribosomes will add by this end. This is called what? Polyribosomes or polysomes. Okay, so these are about protein synthesis. So we completed the replication, transcription and translation. So next class we will discuss about one gene, one enzyme hypothesis and some types of genes. Okay, some types of genes we will discuss in our next class. Then finally we will discuss the operon concept. Okay, operon concept. Two types of operons are there: inducible operon and repressible operon, and we will discuss one example for each inducible and repressible operon. Then we can wind up. Okay, so I hope we required 2 to 3 class to complete this chapter. Okay, thank you.